Topic for this session is microstructure of liver. Liver is surrounded by a fibrous capsule which is called Glissen's capsule. This Glissen's capsule gives inside the liver some trabeculae or septi which divide the inside of the liver into a number of irregular lobules. So these are trabeculae or septi, and along these trabeculae and septi, branches of hepatic artery, portal vein, bile ductules, lymphatics and nerves traverse. So inside of the liver is made up of parenchyma, which forms the 80% of the structure of the liver, and rest is stroma. Parenchyma are the hepatocytes and stroma is the trabeculae containing all these structures and also the tributaries of the hepatic vein. Now, for descriptive purpose, the liver inside is divided into a number of classical hepatic lobules. Sometimes in some pathological conditions, the inside is divided into portal lobules. Third way of describing the interior is by means of liver acini. So first let us consider the classical hepatic lobule. A classical hepatic lobule is a hexagonal structure. It is a three-dimensional structure the center of which is occupied by the central canal. Now this central canal, the central canals, they join to form interlobular veins. So central canal, or I must say central vein, interlobular veins which join to form hepatic veins. These hepatic veins are 5 to 6 in number, and then they drain in inferior vena cava. So each classical hepatic lobular hexagonal structure is 1 mm in diameter. Center is occupied by the central vein, and the 6 corners are occupied by tributary of portal vein, branch of hepatic artery, and a bile ductule. Together these 3 structures are called portal triad. Their branches meet each other at the periphery of a hepatic lobule. So they are all giving vertical and transverse branches. Now if we take section of this thing, then what do we see? We see from above that this is a classical hepatic lobule. I am making 3, each with central vein at the center, portal veins, branches of portal vein at the periphery, along with branch of the hepatic artery and a bile ductule. Now from the center of the hepatic lobule, from the central canal, plates of hepatocytes radiate towards the periphery, and these are called hepatic lamella. One is lamella, many is lamellae. The space between the two lamellae is called lacuna. So if I consider this pen as the central vein, then I can say that the pages of this book are radiating from the central vein to the periphery, and each page is made up of bricks of hepatocytes which are cuboidal in shape. So each page is called lamella, and in between the two pages, this is space. This is space between the two lamella is called lacuna. Like this. Now the lacuna itself is occupied by dilated capillaries called sinusoids, which are lined by endothelial cells. Along with the endothelial cells, they are also lined by special cells called Kupffer cells. These Kupffer cells are phagocytic in nature, which will remove the unwanted material in the blood from the blood within the sinusoids. Outside the sinusoids, the space between the sinusoids and the lamella, this yellow space, is called space of Disse. This space of Disse has another important cells called Ito cells, which help in regeneration of the liver once the tissue is damaged. Now these hepatocytes, which are forming hepatic lamella, they meet with each other around the central canal and form this membrane called inner limiting membrane. And they also meet with each other outside the lobule and form a membrane called outer limiting membrane, which also surrounds the portal triad. So you see, around the portal triad also there is a space, and this space over here, this yellow space over here, it is called space of Moll. This space of Moll is continuous with the space of Disse, and lymphatics starts from the space of Moll. Now the blood in the sinusoids come from the portal triad. The blood which comes to the sinusoids come from two sources. One is from the branches of the portal vein, which brings nutrient rich but toxin rich blood from GIT, and also the blood from the hepatic artery, which is oxygenated. So there's mixing of these two bloods in the sinusoids. Now toxins, now the nutrients and the oxygen, it will be taken up by the hepatocytes, and the carbon dioxide and the toxins, they will be released into the sinusoids. Toxins are actually cleared up by the Kupffer cells, which are present in the lining endothelium of the sinusoids. So once the blood is cleared of the toxins, it reaches the portal, it reaches the central vein. From the central vein, it will reach the hepatic veins and the inferior vena cava. Now 66% of the blood to the liver comes from the portal vein, and only 33%, around 33% comes from the hepatic artery. So this is how the classical hepatic lobule contains the blood from the periphery to the center. Now there is mixing of the blood from one lacunae, it goes to the other, crossing the hepatic lamellae. How it happens? There are gaps between these plates of hepatocytes. So blood from one lacunae goes to other. Even the blood from one lobule goes to other. In humans, the classical hepatic lobule is not clearly visible like this, because here the hepatocytes, they are continuous with each other, and this whole structure, this movement of blood from one lobule to another, from one lacuna to other, is possible because the whole of the liver tissue acts as a spongy tissue. In cirrhosis of liver, when there is fibrosis of these hepatocytes, some hepatocytes are damaged, then what happens? This exchange of blood to the different compartment is lost, and hence this sponginess of the liver is lost.